Today we have Amelia Strom here. Amelia is a PhD student in the MIT Harvard program, mostly working with Jonathan Polimeni and Laura Lewis. I think I've seen abstracts from you for the last 3 years already, so maybe you're already an experienced PhD student. For those who don't know, Amelia cares about kind of tissue dynamics, anything that moves in the brain, so CSF and blood and I guess lymphatic system, and then now also gray matter. Specifically at this year's ISMRM, she had an abstract on kind of showing tissue movements also across cortical depth that got a lot of attention, specifically also in the debriefing sessions of various lab groups that talked about ISMRMs and the highlights. So I was excited when Amelia agreed that she would talk about kind of the movement of gray matter tissue again in this venue. So please share your screen.
Okay, thanks so much for the intro. Can you see it already?
Yes. Yeah.
Okay, great. Okay, yeah, thanks so much for the invitation to speak here. I'm really excited to share the work that I presented at ISMRM this year, quantification of cerebral cortical displacement driven by visual stimulation, along with a lot of added context to really put this in perspective. I'd also like to say at the start, there's very little neuroscience, strictly speaking, in this presentation, but I think the implications of this work are really relevant for high-resolution functional imaging, and I hope that you will all find this both interesting and useful. So before I get started, what do I mean by cortical displacement, and why am I interested in this? So we all know that with an increase in neuronal activity, we get an increase in blood volume, the effect exploited by vaso, for example, but this doesn't happen in isolation. The cortex expands to accommodate this increase in blood volume, and this is hypothesized to coincide with a relative reduction of CSF volume in the region, and also a change in CSF flow. And if you were to peruse my PhD thesis proposal, you would see that this effect on CSF flow is what I'm most interested in measuring and characterizing. So I want to spend a brief moment on that. So as a quick primer for why we're interested in looking at CSF flow changes with sensory stimulation, let's consider this classic understanding of how waste is cleared via CSF and ISF in the brain. We know that CSF can enter the tissue along perivascular spaces, then carry the waste out of the tissue and into the subarachnoid space. But the mechanisms of flow, both within the perivascular spaces and the subarachnoid space, are still unclear. Cardiac pulsations have often been considered as a major driver, but neurovascular coupling also plays a role. So in humans, Zijing Dong and Fusha Wang at the Martinez Center have shown significant changes in CSF flow velocity in the calcarine sulcus following visual stimulation, and Laura Lewis's group showed that there's a significant increase in flow signal as far as the fourth ventricle, also following visual stimulation. And we hope to add to this picture by supplying a measurement of tissue motion that could partially explain these observed CSF flow changes. At the same time, this relative expansion or displacement of the cortex and coincident decrease in CSF volume will also affect fMRI measures. So if we consider this voxel, we can see there's a dynamic partial volume effect, whereby the CSF volume in the voxel decreases between rest and activation. And we often consider partial volume effects in fMRI, especially between gray matter and CSF, but these can also be dynamic, changing over physiological cycles, but also in response to neuronal stimulation. These are data from Sanji King's group showing a decrease in the relative volume of CSF adjacent to cortex in response to sensory stimulation. This is in cats, and multiple groups have shown an increase in cortical thickness in areas of neuronal stimulation, including this work from Polimeni Lab alum Natalia Zaretskaya. And here we see a thickness increase in primary visual cortex during visual stimulation, but not in higher visual areas, V2, V3. So why would we care about these dynamic partial volume effects? Well, they can cause signal fluctuations in fMRI by contributing both to physiological noise and functional contrast, especially because CSF signal is often bright. So for example, in this simulation study from Nick Weiskopf's group, the authors changed the volume and stiffness of a single section, shown in red, of a simulated cortical gyrus, and observed relatively large signal changes in both bold on the left and vaso on the right as the gyrus physically moves, creating a pattern of positive activation on one side and negative activation on the other. And we can also see the same pattern in real bold data from this All That Glitters Is Not Bold paper. These two voxels on either side of a sulcus show signal changes of opposite polarity, which would be consistent with tissue or CSF being displaced back and forth with the same periodicity as the stimulus.
Can I ask about this? Maybe
Yeah, sure.
kind of the summary of those. So I was specifically the Natalia Savetskaya study kind of seemed to suggest that when gray matter dilates, it dilates into white matter. But the Sonji Kim lab kind of suggests the opposite, or is it consistent, or is it just dilating in all directions, or what's the current understanding in the field?
Yeah, that's one thing that I find really interesting also. And yeah, actually show a recent abstract that's found the same thing. So yeah, it's counterintuitive to me, right? That we are thinking about CSF volume changes, but then also seeing the surface displacement only at the white matter, not the PL surface. So I'm not sure. I think it's still unclear, and I think these data hopefully can help provide some more context for that.
Cool.
All right, this effect of CSF volume has been especially discussed in the context of vaso, especially since the contrast in signal magnitude between gray matter and CSF is relatively large. There were a couple influential papers that came from Scouten and Constable at Yale who did this hypercapnia study and showed that the delta CBV maps looked very different before and after accounting for any CSF volume change. However, while this effect seems influential in frontal regions and in auditory cortex, it appears less influential in areas like visual cortex. And then this study on the right measured CSF volume fraction at rest and with visual stimulus and found that this change in CSF fraction is very small or non-existent. But then a few years later, Peter Jezzard's group at Oxford demonstrated a significant decrease in CSF signal fraction with visual stimulation. And as far as I can tell, it seems that the existence of a significant CSF volume change with sensory stimulation in visual areas is still unclear. So that's a quick dive into the CSF volume changes, and I want to touch more on changes in cortical volume as well. So there's been a few studies looking at changes in volume or thickness of the cortex with sensory stimulation, including Natalia's thickness study that I pointed out briefly earlier. Also, Renzo was doing this dynamically almost a decade ago for an ISOMRAM abstract, which is incredible to me. I also enjoyed this paper, or this poster, I should say, from the ISOMRAM joint workshop between the brain function and ultra-high field study groups this year. This is from the Bandettini group, showing a displacement in the white matter surface, but not the PL surface, as we were just talking about. That's the same effect that Natalia noted in her 2022 study. So yeah, this effect is surprising because both of these studies found a displacement at the white matter surface, but not the PL surface. But we just discussed all these changes with CSF volume fraction. So yeah, a little bit unclear and could use some more investigation. I also want to note here how small this effect is. We're looking at a 20 to 25 micron displacement of the cortex, which is about 1% of the overall cortical thickness. So there are some limitations to using a structural measure, cortical thickness, to investigate this cortical displacement and these dynamic partial volume effects. For one, it limits our ability to see layer-specific effects. So if cortical expansion occurs more in deep or superficial layers. I would also say that the temporal dynamics are difficult to figure out with this method, though Renzo might have proved me wrong back in 2016. And finally, these estimations are confounded by known changes in T1 in the cortex with activation due to the increases in blood volume fraction and inflow effects, which would affect signal magnitude and perhaps the estimation of cortical thickness itself. So that brings us to the method I'll be showing you today, which is a motion-encoded stimulated echo EPI technique based on the DENSE pulse sequence, which I will explain shortly. This method is quantifiable, so we are measuring absolute velocity. It's very sensitive to capture very slow velocity motion. It's multi-directional, so we can reveal the degree to which the cortex is moving towards the peel or white matter surface, for example. And it's dynamic to capture the temporal pattern of this effect. And as an added benefit, we can measure CSF flow concurrently to capture both compartments. So I'll spend the rest of this presentation describing our method and how we eventually were able to measure this very subtle cortical motion with a visual stimulation paradigm. So using MRI to measure tissue motion is not a new idea. Researchers have been trying this since the 80s, but most recently there's been an interest in measuring strain with these tissue motion techniques. All these studies that I'm showing you focused almost exclusively on measuring tissue motion over the cardiac cycle, which produces the highest amplitude motion, especially in deep brain structures. That's because with each cardiac cycle, we see a large increase of blood volume in the cranium, which must be coupled with a displacement of volume out of CSF and tissue, since the cranium has a fixed volume. And many of the tissue motion studies over the last couple of decades have used the DENSE pulse sequence that we are adapting now. So DENSE stands for displacement encoding with stimulated echoes. This is essentially an off-label use of a steam diffusion sequence, except we're using a single very low B value. The motion encoding mechanism is analogous to phase contrast, where we have a motion encoding gradient and decoding gradient to capture the change in position of protons between these two gradients. The major difference from phase contrast is that our motion encoding times are much longer, allowing for velocity sensitivities or VENCs of less than 1 millimeter per second, compared with multiple centimeters per second for conventional phase contrast. So this allows for really high sensitivity. The VENC is the maximum velocity that we can measure without aliasing. And to put this in perspective, CSF flow velocities through the fourth ventricle are around 1 to 2 centimeters per second, pretty slow. But if we were to use a VENC of 1 millimeter per second with DENSE, these fourth ventricle velocities are much too fast to be captured, and they'd be aliased many times over. But we need these really low VENCs to look at tissue motion, which has a maximum velocity of around 1 millimeter per second. So this high sensitivity that we get here is absolutely crucial. So because we're working with phase data, there are a lot of factors that we need to keep in mind and correct for that will contribute to our signal. And I just want to briefly review those here. They can be grouped into two buckets: background phase contributions, which are present both when the motion encoding gradients are turned off and when they are turned on, and phase contributions from motion encoding gradients, which are only present when they're turned on. I won't go into detail about how we handle these phase contributions, but one thing I want to note is that this study would not be possible without this Aspire technique that corrects for the head coil channel dependent phase offsets to avoid singularities at 7T, where there's no body coil reference. So that's immensely helpful. Also, the cardiac and respiratory induced motion is much larger than the velocities that we expect with stimulus induced motion. So it's very important to estimate and remove these effectively. And that's precisely what I spent the first couple of years of my PhD focused on. So here's some brief main results for the estimation of cardiac motion, where we have cardiac timing on the horizontal axis and velocity in millimeters per second on the vertical axis. First in the brainstem with the velocity plot on the left and displacement on the right. So the native measurement of this method is velocity. Again, since the motion encoding mechanism is really similar to phase contrast. But we can quantify displacement by simply taking a cumulative integral of the velocity over time. So we can go back and forth between these measures. We can also repeat this in the visual cortex, which has much less cardiac locked motion, but we can still see robust cardiac locked velocity changes in the cortex, but of course with a much smaller amplitude compared to the brainstem. For respiratory motion, we can measure both the phase induced by changes in the B0 field with respiration and physical tissue displacement. So first, I'm showing the phase change associated with full inspiration for individual subjects in color and a subject average in black, and each column here represents a different direction of motion encoding. This phase change includes both phase induced by B0 changes and physical tissue motion. So we can measure the B0 induced phase for each subject using the B equals 0 images, then subtract this to estimate tissue displacement, which is shown here. And we can see that the greatest respiratory motion is seen in the Z direction, as we would expect with this. Okay, so now we have all the factors that we need to process these dense phase data and correct for phase contributions. So it's time to think about bringing sensory stimulation into the mix. And that brings us to my favorite feature of this method, which is that the magnitude value data are bold weighted. So far, we've been talking about the phase value data, which are crucial for velocity measurement, of course, but the magnitude of dense is T2 weighted. We are also using a very low B value, so there's negligible diffusion contrast, and we're really capturing bold both with and without these motion encoding gradients. Also, since it's T2 weighted, it has microvascular weighting, similar to spin echo bold. Of course, this is a stimulated echo sequence, not spin echo. So the T2 and microvascular weighting will be slightly different. Our colleague Avery Berman did these simulations showing vessel radius on the horizontal axis to demonstrate that this stimulated echo bold indeed has similar weighting towards the microvasculature as spin echo. We also showed this in practice at last year's ISMRM. So on the left with the magnitude value data, we can see a bold effect with the visual stimulus, whereas on the right, we can use the phase from the same data in the same ROI to look at cardiac locked velocity. Note again that this is stimulated echo bold, which is not often used, to say the least. The CNR is much lower than gradient echo or even spin echo bold, but we really need to use the stimulated echo bold for this because we need the sensitivity of the stimulated echo for tissue motion quantification. So it's a trade-off. And that brings us to the study that I presented this year, where we wanted to use this method to measure cortical displacement locked to a visual stimulus. So for the study, we acquired data in 7 participants at 7 Tesla during a visual stimulation paradigm with cardiac and respiratory monitoring. Our voxel size is 2 millimeters isotropic, and our velocity sensitivity is 0.8 millimeters per second. Note that with the very sensitive motion encoding, we can measure velocities that are much smaller than this voxel size. I also want to emphasize how long these data take to acquire. A full scan session with all the required acquisitions takes about 95 minutes on a good day. So acquisition time is certainly a limitation for this data set. And you can see many of the pre-processing steps that I mentioned earlier, which again are vital here. So first, with this method, we're looking concurrently at bold and tissue velocity. So first, we're using the magnitude to capture the bold response, and we can see stimulated echo bold responses in the dense data within individuals as expected, though with substantially less sensitivity than gradient echo bold, and you can even see the difference in the number of trials between these two. For motion in the phase data, we would expect that cortical tissue velocity would be highest when blood volume changes. So that coincides with the derivative of the bold response. We expect that the spatial pattern would vary based on cortical folds. So the velocity vectors that we calculate from the phase data would be dependent on cortical orientation. To capture the temporal pattern of motion, we first computed the norm of motion vectors within each voxel using the trial averages in each of our 3 directions. So this analysis is essentially agnostic to the direction of the vector. Then we average across a visual ROI. And this analysis answers the question, at what time points during the stimulus trial is the amplitude of motion greatest? Or when is the tissue moving the most? So here I'm showing these average motion vector norms at increasing thresholds for bold signal based on our independently acquired gradient echo bold functional localizer. And even with this liberal threshold, we see greater motion at both the onset and offset of the visual stimulus as expected. And as we increase the threshold, we see greater motion at these same peaks. If we then convert these velocities to displacement, these peaks correspond to a displacement of 31 and 34 microns respectively, which is slightly larger than previous estimates of a cortical thickness increase of 20 to 25 microns. I also compared this to a control region, the temporal cortex, which doesn't have significant bold signal and shouldn't show significant cortical motion locked to the stimulus, and we don't see the same pattern here as we hypothesized. We can also compare the time course to the derivative of the concurrently acquired stimulated echo bold signal that comes from the magnitude value data. So here I'm taking the absolute value of this stimulated echo bold derivative to match our vector norm metric for easy comparison. And we can see that the temporal pattern of each of these match qualitatively, again, as we would hypothesize. Next, we can compare patterns in the tissue and CSF, since these populations should have different motion patterns locked to the stimulus. So first, we took a small subset of the visual ROI with a high contribution of CSF, and noticed that the CSF vector norm is much higher than that of tissue. And this is an important observation because our voxel size is relatively large at 2 millimeter isotropic, so many of our cortical voxels likely contain some CSF signal that may affect both the magnitude and direction of their measured motion vectors. So we would hypothesize that motion vector magnitude and direction differ between tissue and CSF. Motion is faster in CSF compared to tissue, as we just saw in the previous figure. So voxels with greater CSF contribution should have larger vector norms. For differences in direction between tissue and CSF, we can use the cortical surface normal as a point of reference, since we know that the absolute direction of our motion vectors will vary based on the cortical folding. And we hypothesize that cortical tissue moves parallel to the surface normal, consistent with an expansion of the cortex, while the CSF moves mostly perpendicular to the surface normal, corresponding to flow along the cortical surface. To label voxels as having more or less CSF contribution, we can look across cortical depths using a surface-based analysis. And we would hypothesize that voxels at the peel surface with greater CSF contribution will show larger motion vector norms compared to deeper voxels. For this surface-based analysis, we had to get a little bit creative since our voxel size is relatively large, and you can clearly see that with this surface overlaid on the magnitude valued dense image. So we decided to create ROIs intersecting the white matter, mid-gray, or peel surfaces shown here. Despite these large voxels, there's relatively little overlap between these ROIs, so I'm treating them as separate entities. Then we masked these to only include voxels within the visual ROI for this analysis. So first, I will look at motion vector norms, similar to previous results that I showed, but now across cortical depths. And with these results, we can see that the motion vector norms look pretty similar across these ROIs, but they're generally, though not significantly higher at the peel surface compared to white matter and mid-gray, which is the direction that we would expect given the larger degree of fast-moving CSF in these peel voxels. Note that the observation of motion in voxels far from the peel surface, especially at this white matter surface, suggests that our detected motion reflects not simply just CSF motion, but tissue motion as well, since these voxels contain little or no CSF contribution. So next, we wanted to see if the direction of the motion vectors varied across depths, or in other words, with the degree of CSF contribution. The most straightforward way to look at this would be to calculate angles relative to the cortical surface normal, since we hypothesized that tissue and CSF would be moving in different directions relative to the cortical surface. But this proved to be incredibly difficult and impossible to interpret, since these data have quite low SNR. I also tried to look at individual voxels or groups of voxels with clear cortical orientations to get any sense of direction. But again, these data are just too noisy. So instead, we decided to simplify it considerably by looking at the degree to which our motion vectors are parallel or perpendicular to the cortical surface by using the dot product and norm of the cross product, respectively. We would expect that voxels with less CSF contribution deeper into the cortex should be more parallel to the surface normal, whereas voxels with more CSF contribution at the peel surface should be more perpendicular. So we computed these measures at each of the two peaks that we identified with the vector norms. And when we look at the dot product, we don't see any evidence for parallel directed motion for any of the cortical depths. When we look at the cross product norm for degree of perpendicular, we can see some evidence for voxels at the peel surface being more perpendicular than baseline, which might suggest that we are perhaps capturing some CSF motion there, but the effect size here is very, very small. So ultimately, the vector norm effect is quite robust, but the vector direction analysis shows little preferential direction. And this might be due to the sensitivity of our method, since it's difficult to look at directions sort of individually instead of averaged together. I think that analyzing the vector direction instead of its norm is somewhat akin to transitioning from a mean diffusivity analysis to a tractography analysis. Just makes things more difficult. And also, our relatively large voxels reduce our spatial specificity, like in terms of separating tissue from CSF. And it could also be that the underlying pattern of motion doesn't match our hypothesis that we tested for the direction analysis. So next steps, we plan to implement a line scan version of DENSE, since understanding the pattern of tissue motion across layers is difficult without high spatial resolution along the cortex due to the ambiguities arising from partial volume effects. So we can consider a line oriented perpendicular to the calcarine sulcus, covering both cortex and CSF. And again, we expect that cortical motion will be oriented parallel to the surface normal with some pattern or gradient across cortical depth, whereas CSF will likely be oriented perpendicular to the surface normal. And with our 2 millimeter voxels that I've just shown, it's very challenging to find any spatial specificity for gray matter or CSF, or reveal any pattern across depths, because there are two populations, at least gray matter and CSF, that are likely moving in different ways, but are being averaged together in these large voxels. With a line scan acquisition, though, we'll have much better resolution to differentiate tissue and CSF, and we'll also have access to patterns across cortical depths. So this study will allow us to capture a layer-specific measurement of cortical displacement with stimulation. Our lab has previously used line scan diffusion MRI to look at microstructural differences across cortical layers in humans, and improving line scan methods is an active area of work for us. So we think this is actually a feasible study that we hope to try in the next few months. To finish up here, I want to demonstrate how this cortical displacement could manifest in your high resolution fMRI data. So I made some calculations based on the effect sizes that I've shown you today. So if we were to assume an 800 micron, essentially one-dimensional voxel at an initial partial volume of 50% CSF and 50% tissue, and a 20 micron displacement of the cortex towards the CSF, this would result in a 2.5% volume change. Next, we have to assume some signal magnitude, which I have arbitrarily set to CSF with double the magnitude of tissue. And these parameters taken together would result in a 0.8% signal decrease coinciding with the stimulus, simply due to this dynamic partial volume effect. I also repeated this for a more aspirational voxel size of 300 microns, which would result in a 2.3% signal decrease. But this dynamic partial volume effect would also appear in the noise. So we can consider the repeated acquisition of a voxel with 50% CSF and 50% tissue at baseline. And the tissue would displace to different locations depending on the cardiac timing of the acquisition, leading to a dynamic partial volume effect due to cardiac motion. So there's variable cardiac motion induced noise depending on the voxel's acquisition time in the cardiac cycle. And the amplitude of this displacement is variable depending on location and timing, but can range from about 10 to 40 microns, so with a similar amplitude to the stimulus locked motion. So in summary, one of the reasons we are interested in measuring cortical displacement is because neurovascular coupling might play a role in CSF flow and waste clearance, and tissue motion might be a part of that story. I've shown that we can acquire stimulated echo bold fMRI concurrently with tissue motion quantification, and that we observe cortical tissue and CSF motion at the onset and offset of a visual stimulus. The current method has limitations, most notably the large voxels and long acquisition times required due to low SNR. I hope that you leave this presentation with appreciation for how dynamic partial volume effects caused by cortical displacement might contribute to fMRI signals, both in the noise caused by cardiac motion and in stimulus-evoked signal caused by the local blood volume increase. And these effects become more prominent with smaller voxels, in voxels at the peel surface, and in acquisitions with a large contrast in signal magnitude between CSF and tissue. So I want to thank my advisors, John Polimeni and Laura Lewis, all the people that helped make this project happen, and thanks to you all for listening.
All right, thanks so much. That's really fascinating. Like, the sensitivity is enormous. Like, it's maybe surprising, right, that you can see kind of motion sensitive, these tiny signals with motion sensitive sequences, whereas kind of partial volume based methods kind of could really benefit from the gray matter CSF contrast to noise ratio that can be very high. But you see kind of more with motion sensitive sequences, specifically across time, which is really impressive. So I have a couple of questions, but maybe before I start, is there anybody else who has a specific question? Yeah, Farouk.
Thanks for the nice talk, Amelia. I had two questions in mind while listening. First one is that you mentioned that it is unclear whether the motion happens mostly towards white matter or to the outside towards CSF. Is this correct? That I heard it correctly, right?
Yeah. This comes from these cortical thickness studies that use the peel and white matter surfaces with just a structural scan during a usually just a structural scan during the visual stimulus versus at rest. And you can sort of quantify how much the surfaces are moving, either the peel or the white matter. And two studies that I showed today found that change at the white matter surface, but not the peel surface with that technique.
So about that part, I was wondering if there is any like separation with regards to gyri and sulci in the brain, like thinking of the overall geometry of the brain, whether like this is like a curiosity question, like whether you would expect it to be in a certain way given how much white matter material is underneath the gray matter tissue and things like that. Do people talk about these things in that literature?
Oh, that's a great point. I haven't seen that discussed, but yeah, I imagine that curvature would affect sort of mechanically where the tissue can move to, depending on the sort of resistance patterns with different curvature. So I haven't seen that discussed, but that's a really interesting point.
I see. Then it might be interesting to look at just in a very crude way, like sulci and gyri, and maybe just a straight pieces of the cortex, like separately, maybe. Like you mentioned that you were struggling to make a very local analysis, right?
Yeah.
Versus like ROI, maybe like even like a larger ROI could be just looking at the 3 types of general curvature classifications.
Yeah, that's really interesting. Thanks.
The other question I had in mind was that, so you overlaid a story about the CSF and the tissue partial voluming. While I was listening to that, I was thinking, well, what's going on with the veins? Like if the overall blood volume is larger, right, veins will become larger as well, I think, eventually. And the veins have even a lower signal than the gray matter tissue, then the partial voluming between the veins and the CSF will be really, really strong. So I was just wondering, like, in your thinking, like especially with regards to the pile surface, do you position the vessels there, like veins and arteries, of course, like in this like partial volume equation?
Yeah, not right now. I don't think we have the data to look at that yet, but that's something that we're thinking about. Yeah, with the expansion of both the veins and the arteries and how that affects motion around them. I think it's a really interesting sort of avenue that we'd love to take a look at, but we just don't have the data for it just yet. But yeah, I totally agree with you.
Okay, thank you.
Maybe I have a question about veins as well. So based on my experience, my own experience with non-bold contrasts, such as vaso, I found that kind of one common theme that I always encounter is that bold is very hard to get rid of, specifically as you go to kind of more sensitive sequences. So in the STEAM sequence, you're, as far as I understand, you're mostly sensitive to kind of T2-like contrasts. There might be a bit of R1 prime, R2 prime from the EPI readout, but yeah, at 7 Tesla, like for spin echo sequences, the intravascular bold is like still there to some degree, right? It's not completely gone. So would you think that kind of, we know that oxygenation changes specifically intravascularly, but also kind of dependent on vessel orientation, would result in the phase differences, right? So there would be a kind of susceptibility change during activity and rest that affects your phase data. Is that too small that you cannot see it in your phase data? Or how, maybe the general question is, do you think there's a possibility that you still have bold contamination in your phase data that you interpret as kind of velocity data mostly?
That's a really interesting point. I only thought about it in terms of the bold changing the magnitude, which changes the sort of partial volume contributions. But I hadn't thought about the susceptibility change directly affecting the phase. That's a really, really good point. And I haven't even looked into that, but thanks.
Maybe another question on vaso. Like, I think maybe it's more a comment. Like in vaso, there are several kind of variants in there, and specifically in the early days when people kind of were trying to do whole brain vaso, they were doing this magic vaso, the Scout and Constable sequences, which had these kind of repeated inversions. And then also, I think in the Weisskopf study, they use a kind of synthetic vaso where they're not considering steady state. So CSF in that figure was extremely bright, which I've never seen in practice. So maybe in the last decade or so, since 2012, the vaso variants have changed quite a bit in the sense that now the inversion times are usually longer because you're assuming a different steady state of the tissue versus the kind of the flowing blood, and therefore the CSF to gray matter signal is actually getting quite similar. So CSF is getting very, very dark for those kind of longer inversion times in steady state, at least. So I think kind of the implications that you mentioned on the second to last slide or so, I think there's a lot of degree of freedom. And you mentioned, I think you simulated maybe if you go one slide back, the tissue contrast would be 100 and tissue would be 50 and then CSF would be 100. So if you can kind of level those, like for example, in things like SSSI vaso where they're very, very similar, then you can kind of even tune your sequence to be more or less sensitive to that. And there's very often we have degrees of freedom for that, like the flip angle, for example, in bold, or if you use 2D or 3D, you can equalize those contrasts quite a bit and then sensitize it to those kind of partial volume changes or make you less sensitive to it. But I wouldn't say that kind of generally kind of vaso is very sensitive to that because it depends on kind of on the specific variant that you're using with a given flip angle.
Yeah, good point. This is really like a sort of worst case scenario type of thing. And if your magnitudes are the same between them, then this wouldn't really contribute at all. So yeah, but just something to keep in mind if you're using a sequence that doesn't have that by default, that this might contribute. But yeah.
I would have another question if nobody else has one. Oh yes, Tyler.
Yeah, really, really beautiful talk. I was curious a little bit about the kind of future plans for line scanning, just because like since you're doing phase sensitivity, obviously having non-isotropic voxels or anisotropic voxels means that like your sensitivity is going to be different in different directions. Do you have any plans for like, do you need to do more sampling in the motion encoding along gray matter rather than through it, etc.? Or like, have you thought through like what the sampling patterns are going to need to be in order to kind of accommodate the fact that the phase is going to accumulate in different directions?
I haven't gotten that far yet, but yeah, definitely something to keep in mind. I appreciate the suggestion. All this, the nitty-gritties of the line scan are things that I'm not familiar with myself just yet. So I'm really relying on our lab members for that expertise. And yeah, that's certainly something that we could consider.
Very cool. Yeah, it might turn out that like the sensitivity that you have is actually best for the direction that you're wanting to go. So it'll be really interesting to see how that turns out.
Yeah, I agree.
Burak.
Hey, it was a great talk. I want your idea about BCG motion. I mean, you haven't mentioned about that. After the QRS peak, there's like a blood rush to the brain, and then it just moves the whole brain, including the skull, about, I don't know, 50, 100 micrometers, more than the displacement you try to measure. Do you think it will affect any 20 micrometers that you mentioned that confound that cortical measures depending on the cycle?
I'm so glad you asked about this. I did a small project looking into this specific effect by using the, well, a couple different ways, by using the scalp phase. So I can see that, sort of BCG effect from the scalp. And then I've also done like sort of a single slice sagittal with a really fast TR to see this in a different way and saw something similar. Yeah, so I'm really glad you asked about it. Within the tissue, those effects would be captured in the same way as I do my cardiac motion estimation. So they'd be removed the same way that real like non-rigid displacement of the tissue would be removed. But yeah, I love thinking about that, and I'm so glad you brought it up.
And regarding the analysis, also the desculling the brain or like doing the registration, that may affect also that globally, right? Volume to volume.
Yeah, definitely. So one thing with like with motion correction, we're really just looking at the magnitude value data and just doing sort of a classic motion correction. And I haven't implemented something to take into account this rigid head motion that affects the phase. That's a lot more, it's a more difficult problem to tackle, but I think the scalp measures give us a half decent sort of non-rigid head motion estimation. But so far, I've had wonderful subjects that hasn't been a concern just yet.
Thank you.
I would have an understanding question. So you mentioned in your physiological noise correction that you have data without any diffusion gradients, and you use those for. So you're acquiring always kind of everyone 4 times. Is that right? Like 3 different directions and then no diffusion.
Yeah.
Or maybe with B0 you were referring to kind of the B0 field or B value 0. I wasn't sure.
Yeah, it's they're all so similar terminology. Yeah, so the way that I have it set up is each run is in a single direction, but I start with 10 frames of B equals 0 images without any motion encoding gradients. So for every run, regardless of direction, I have those 10 frames without any motion encoding. So I can use that to correct for background phase offsets for the run, but then also use them in combination to look at respiratory B0 changes, for example. Does that answer your question?
Oh, yes. And that's very smart. And then you, from the navigators of each TR of the diffusion weighted scans, you use that for removing the respiration. Because you need to have it in real time, do you?
Well, I had those essentially 90 frames in total, and I have respiratory monitoring during that time, so I can sort of concatenate all those together, reference back to the respiratory trace and estimate that way.
Okay, makes sense. Okay, cool.
Great.
Like looking at this slide, and maybe as a last comment, I'm really excited about the line scanning because kind of you mentioned that you're motivated that you want to look at motion because blood volume alone kind of is never happening in isolation, as you said, right? Because when blood volume increases, something else needs to decrease, and it's a complicated story. For motion, it might be not too different, right? When you have motion in one direction, as long as the entire brain isn't moving, kind of something needs to flow in the other direction. So I guess you need to be at the right spatial scale where you have coherent motion in one direction, but then kind of in a different location, then the motion is kind of flowing back as long as the entire brain is not moving. And then I guess key here will be resolution. And then you can, I guess, have a ultimate answer to the question of kind of if when blood volume increases, if basically gray matter swells and you have motion in both directions, opposite directions, which you might average out with your 2 millimeter boxes, right? But with light scanning, you could see if they move in different directions.
Yeah, I'm also really looking forward to all that. I think, this is something I've been wanting to do basically since I started because of all the just fine spatial scales that these things are happening on. So yeah, I mean, we'll see how it goes, and there's a ton of nitty-gritties to go into in terms of all the different directions things could flow into, the timing of each of those. So looking forward to it, really.
Tyler, you switched on your video again. Did you want to say something?
No, I mean, I would say the same thing. I'm excited for this part of it just because like it's going to take solving some of the problems that already exist for line scanning in general to be able to do that project. But there's some of the more interesting problems, like how do navigators work? How do you know that the brain is not all moving? And simultaneously, it's just, like what Farouk brought up, line scanning is limited to one portion of the vertices that you can really use. And so it can answer the question under certain circumstances, and finding solutions for how to solve it under gyri or sulky will be really interesting too. Yeah, it's very exciting work.
Thanks.
All right, if there are no further questions, thank you, Amelia. So in 2 weeks, we're going to meet again, and that time we will hear a presentation on visual and basic cognitive neuroscience from Shaheen Nasser. See you in 2 weeks.
Thanks, everyone.